Thank you so much, Bob. It's a pleasure to be here this morning for me here over in Cleveland, Ohio, where the company Trailhead Biosystems is located. Such a pleasure to join in on the Reprocell webinar series. We are focused on making specialized human cells. I'm going to talk today about a specific technology that the company has developed over a number of years, and how it can be used to accelerate the differentiation, the directed differentiation of induced pluripotent stem cells to the more specialized fate. Today, before I start, the disclaimer: I am a board member. I'm also the CEO and CSO of the company, and of course a shareholder in Trailhead Biosystems. The company is, as I said, located in Cleveland. We operate out of a facility that we wholly own. We have generated this, as I will show you later, with laboratory facilities, tissue culture, tissue cell production, and bioinformatics, and some other things. Our motto is a very audacious statement: biology controlled, and it's not something you just make. So I hope that today that I can explain how we're able to make this statement. It all has to do with mathematics. Now, returning to the cells before we get there, as you all know, you wouldn't be here today unless you have an interest in induced pluripotent stem cells. You may be working with Reprocell products. You may be doing directed differentiation yourself, or in other ways or forms actually use induced pluripotent stem cells in your daily work. It's sort of common knowledge that now, after the initial discovery by Yamanaka and his colleagues, that one can reprogram an adult cell into a cell that is reminiscent, actually almost identical, at least very similar, to the inner cell mass of the embryo. Those cells, when reprogrammed, are stable, they are immortal, but they're also not really very useful. They don't have many specialized features. So in order to get the utility increased in our iPSC cultures, we must differentiate them. And now that's a process that is very, very hard. A lot of effort is going into this, as I'll explain, and most of this is referred to as directed differentiation, essentially seeking to emulate the processes that would usually happen in the embryo. But as we don't really know yet the built print of the human organism, how are we going to accurately and efficiently steer cells to the specialized cell fate? This is what this presentation here is all about, and it is the basis of our company, Trailhead Biosystems. The problem as it exists today, when we are attempting to do this at lab benches around the world, is that the process is mainly manually done. It is mainly done by hypothesis-driven research, individual testing of a set number of inputs, and seeing what works, and then gradually build a protocol up. That is an iterative process of science done in many labs, attempting to make an improvement to a previously published protocol, rarely touching the first stages of the protocol. That leads to generally a lack of purity, a lack of functionality, and is very, very, very difficult to industrially manufacture, because the process understanding that sits underneath that is really not captured in that discovery process. And currently, just taking the NIH funding into this area, it is very clear that we're nowhere near the limit of spending money trying to make better cells. If you look at embryonic stem cell research in general, including that of induced pluripotent stem cell, that of course began to ramp up after the original discovery of Yamanaka in 2018, then we are now, just from the NIH budget, spending more than $2 billion annually on pluripotent stem cell work. Now, directed differentiation still carries a big burden here, where people are attempting to make cells, where there are more than $400 million currently spent in projects that are supported towards directed differentiation, split into around 800 individual projects. So it's not for lack of trying, but and the cells are certainly useful, but again, we usually run into trouble when wanting to industrialize or clinically test the cells. I'm here today to talk about what we did starting around 10 years ago, where we replaced this manual discovery process with a machine enablement. We are now capable of exploring a high-dimensional space of signaling inputs that then control cell fate. And we're always looking for combinations of signaling factors, just as the factors in the embryo are controlling cell fates. We're not trying to cheat ourselves through, for instance, overexpression transcription factors or other things that we can steer the fate through a bioinformatics-driven approach. This is essentially a normal induction process using particular molecules that emulate or are exactly identical to those operating in the embryo. When we do it, we find that we achieve much better cell purity, and we're then taking these process control parameters into manufacturing, seeking to build more and actually go to market with these products. The technology I'm talking about is what we refer to as HD-DoE, high dimensional design of experiments. It is simply a dimensional amplification of standard design of experiments, which is ubiquitous in all manufacturing industries across the world. However, it hasn't really been applied at the dimensionality that we're at, because biology is a tad more complex than typical physical chemistry and engineering problematics. To do it, we've developed some proprietary tools to perform these experimentations. They're all done through the computerized experimental designs, and they're robotically executed. It's the only way to do it because the number of runs are simply too vast to do by hand. When we generate the data, we generate our own data, and we mine these data for the protocol inputs that we need. In other words, we don't really need to go out and look at other people's data to build the protocols. It generates a data-driven and unbiased determination of what is mathematically defined as critical process parameters, the biological ones that underpin that of control of cell fate. So if you look at it this way, mathematics is a very interesting thing for a biologist because we tend to shy away from that. But here it's really showing its power. We are able to compress what otherwise would have been a very large testing space, represented to the left here, as what would have been necessary if you're doing something called a full factorial testing in 12 dimensions. The number of runs that you can see would be 2 to the power of the number of dimensions. In other words, more than 4,000 individual runs. That would be very costly to do. And it's not necessary because we're able to use our mathematical compressions to test less than that, while we still retain the ability to identify the primary interactions amongst the factors that we test. So that reduces the cost, and it certainly reduces the time. Without going into depth with exactly how it works, because I can't really do that, and secondly, I also don't have the time, the output of the data allows us to generate what is called an effector response matrix. The matrix is essentially how does a set number of genes operate in response to X number of effectors that is seeking to control it. And we can hereby then extract what is the regulatory imprint on a gene. We can find which factors that, when provided alone or in particular in combination, will up or down regulate the gene. We can then arrange all these genes that we've measured in such an experiment according to how they're regulated, and group them according to regulatory similarity. So when two genes get together in a plot like this, it simply means that they're subjected to the same signaling inputs and the combination thereof. And in this situation, we have a fate split in what's called the medial ganglionic eminence progenitor of the forebrain that generates two separate interneurons, one being parvalbumin positive and the other one being a somatostatinergic neuron that is complementary to this. The culture simply can't coexist between the two. It'll either go to one or the other, and we can here identify which combinations of recipes we need. This allows us to mine the recipes in a very comprehensive way as we move through the high-dimensional space. This allows us then to build directed differentiation protocols. These protocols are always built by us. We always build them from the ground stage all the way to the end. and we do not copy literature. We will test literature inputs. We will read the papers on how to build this or that cell. We will extract the input components and we will test them. And we will also test other things that we have from our historical data or other ideas that we can test from our library of compounds. And then we would mine that stage-wise as we jump between each stage. So it's a lot of work, and you can more or less say that it represents to a certain extent that type of work that I just mentioned on that NIH funding slide, that we're sort of dueling with the company's technology in context with that of all the world's directed to differentiation. But we feel like we need to build these protocols again because we don't have a definition that, for instance, if you're doing an islet cell differentiation, that you have to use activin and Wnt in the first stage just to do a definitive endoderm. We don't do that. For instance, same argument, there is no reason, and we have found that it is not necessary nor optimal to use a dual SMAD induction strategy on stage 1 for any of our interneurons or other neurons. That method simply generates a very vast neuroectodermal field for which the protocol is not really steered well enough onto the first stage that you then need for the next stage. So in other words, there is an argument that you want to develop these protocols with an understanding of the critical inputs and try to identify that before you actually venture too hard into a scale-up context. We've built now many protocols, and we have around 25 different cell programs strewn around the germ layers in the embryo. And I do think that we have the highest number of active cell programs of any company in this space, and this is because the technology provides for that. You can sometimes see phase splits like we are seeing here with our oligodendrocytes and a cell type called the VLMC that we've now produced and will release to market. The VLMC is an abbreviation for the name vascular leptomeningeal cell, which is actually a cell that participates on the neural side in the blood-brain barrier. But I could talk a lot about that, and I don't have time to do that. So I will suggest that you go to our website and follow our news, subscribe to our LinkedIn, and follow the company for the information as we go forward onto the various cell programs. We're very proud, before I jump on, that we've discovered a way to make very efficient hematopoietic stem cells and hematopoietic progenitor cells that have full muscle lineage competency that we can manufacture at scale, because it has immediately opened up the generation not just for HSCs, but also for descendant, say, monocytes, macrophages, or the myeloid cells, and including the lymphoid ones, erythrocytes and megakaryocytes as examples. So we have, of course, initially focused on the cells that have the highest therapeutic opportunity and need. Those are, for instance, pancreatic islet cells for type 1 diabetes. Another one is, of course, the A9 subtype dopaminergic neurons shown here. For Parkinson's disease. Both diseases are particularly ripe for cell-based therapy. And as you know, companies like Vertex, Novo Nordisk, and others, and Bayer, have ongoing cell therapy programs in the clinical stage right now, testing iPSC-derived cells or embryonic stem cells. We're not really feeling that the protocols out there necessarily may be the best. So we're seeking to build a better cell protocol up. Every time I mention these protocols, I refer to them as sort of second generation, because they're the only ones built with this methodology. They're built to achieve the purity and function. And of course, as I said, every time we start from scratch and then move through our development schema. And by the way, for those interested, we're very strong subscribers to what's called quality by design, which is an overall product development schema that is also used across other industries and is particularly well suited to be applied here. When we have made the cells, we of course need to also show that the cells are the right type. How do we do that? Well, now we're back to the standard science. We are, of course, using the standard methods of sequencing. We're using qPCR, various molecular methods. We're using a lot of immunofluorescence, particularly for the brain cells and the neuroectodermal cells. For the blood cells, flow cytometry is a hallmark of technology. And there are other functional assays that you need to apply just to show that the cells are what you want them seek to build. I can't go through these programs to any large detail today, simply due to the lack of time. When we've made a cell, we would like to go out and compare how does it look with others. We can go to publications. Our scientists are looking at the standard assays, and we, in some situations, are able to compare with some companies' offerings in the market. So, for instance, the dopaminergic neurons here can be acquired from other companies, and we have one producer here for which we sourced in the cell, and wanted to compare our own cells with that product. We follow instructions according to their the specifications, and then here is the performance of an ELISA analysis of the amount of dopamine produced by blue, our dopaminergic neurons, and then the other companies. And we were surprised to see that we actually couldn't really see much at all. But it's not that of the competitive product, but there is dopamine produced. You just have to sort of look closer at the bottom of the axis. So we also know that the percentage of dopaminergic neurons here in this culture here reaches between 60 to 80%, which we think is, we can see from literature, is quite a bit higher than typically the sub-single-digit percentage that others often claim. So we are, of course, interested in obtaining successful partnering with this cell product. And we are therefore negotiating with therapeutic developers to sort of piggyback and move this particular protocol into patients for testing. The argument being that when the cells have a higher lineage-specific fit to the A9, the SOC6 expressing dopaminergic neuron, which is the only one that's lost in the Parkinson's brain, then you should expect a better clinical outcome. And as we talk to therapy developers, they come to us and say, well, can your cells be actually going forward in a clinical context? And then most of the stuff that we were doing to build this protocol up was actually on an NIH repository line, which is not consented or commercially cited for that purpose, nor has it been CGMP produced. So it gives us a little bit of a pause in discussions at that point to say, actually, we must, so we can go back and say, well, do you have the line? And maybe they do, or maybe they don't. But this is where the opportunity to work with a company like Reprocell has really emerged well for us, because Reprocell sits at that stage to source, create cell products of different iPSC sources with the RNA-based iPSC generation, and there is a sideline both for clinic and commercial use. And it is for that reason that we began to shift over, and we, of course, then seek to evaluate our protocol on this other iPSC line. We, by the way, have been very happy when we shifted between iPSC lines, so we have less of a founder-founder or donor-donor effect on the source material. And so I'm going to show a little bit of data here on using Reprocell cultures in the context of our dopaminergic protocol that was developed on the CR01 or the NCRM01 line. And this is just a snapshot of comparative data. But if we follow at a given stage in the protocol when the cells should have matured, the Reprocell line is a little bit slower out the gate, but otherwise follow the protocol instructions quite well. We do see that we can attain the SOC6 expression. We can attain actually even more tyrosine hydroxylase expression. And there is an even further reduction on the stray fate for astrocytes, for instance, as evidenced by GFAP. So we were pretty happy with that. If we look on the percentage of conversion as we move into the stage 4 of the protocol, we're also pretty pleased with the number of neuronal cells emerging, and also with the fact that we may be a little bit below on what we had on the CR, but it's still a very good conversion into the dopaminergic fate with the Reprocell line, and not that many CD44 glial either progenitors or more mature cells. on the non-desired lineage. So hold on, I think I went the wrong way. Staining for the cells, here's a couple of pictures. I do believe that they're in two different magnifications, but it's the same staining pattern for tyrosine hydroxylase and that of TOUCH1. And again, we can see the bundles of projection neurons in both cultures, and there's plenty of abundance of our desired marker, tyrosine hydroxylase. So by and large, we're fairly pleased with the performance of this Reprocell line using the other protocol with no modifications done in the protocol to make it fit that line. If I move outside the specific Parkinson's target area, we can look at the human organism, and there are around 500, more than that actually, according to the single cell specialists out there. But we know we have at least more than 500 distinct cell states residing within the different tissues and organs across human biology. And we are currently at around 25 cell programs covering this and that there. So we're 5% coverage, which I'm very pleased to see. We have internally in the company split this work into 3 different areas according to the germ layer, and that is ectodermal programs, endodermal programs, and mesodermal programs. And the ectodermal programs mainly focus on the brain, and we've mainly focused on forebrain and midbrain for now. We're now moving into the non-neural ectoderm as well with a new program to make skin. And we would like to move into the spinal cord, but currently don't have really much of a spinal cord program. We need to staff in for that. Endodermal programs is mainly focused only on 2 cells at current, and that is the pancreatic islet cells as number 1, which was our legacy program, and then the hepatocytes, which has really moved super fast forward since the end of last year, where I would love to show you data on that, but you have to follow us towards the release of hepatocytes, both for research use in hepatic toxicity assays for drug discovery, but certainly also for the use in a therapy context towards end-stage liver disease. Aside from that, you can see there's a lot of individual programs on blood, because after you've made the HPCs, you got the gates open to drive into individual blood lineages. We are talking to cell therapy developers in the following way. We've built a therapeutic cell program portfolio up where we subsegment the various data elements that we have for the various cells. This is just the front page of some of these. For Parkinson's disease, the matching cell is the A9 subtype. Dopaminergic neurons for Huntington's disease, it is the DRD2 medium spiny neurons of the lateral ganglionic eminence. Those are the ones that are lost in Huntington's. We have a nice program on that. We have various interneurons that address issues in new psychiatric disease like autism and epilepsy and schizophrenia. We have oligodendrocytes and now also astrocytes, and the oligodendrocytes are interesting because of MS and various leukodystrophies. The VLMCs that I was talking about is a parasite-like cell of the brain that participates in the blood-brain barrier and should be used for studies of neuroinflammatory diseases and probably also a lot of other things. Then we have endothelial cells that we sell. These can be used for various elements of vascular disease biology. And then we've made HSCs. We're seeking to prove that they can graft. There was a beautiful paper by the group of Elefanti in Australia related to a successful grafting of iPSC-derived HSCs recently, and we're seeking to prove this on our cells as well. But we're using these cells already to make descendant lineage cells, like for instance macrophages and monocytes and dendritic cells. We're also making red blood cells, as you can see from the little spin down from a reactor there. Those are hemoglobin containing erythrocytes. And then we're proud to have our diabetes program, which is increasingly moving towards functional maturation of the beta cells. This is a lot of cells. But the interesting thing is that we can quickly make a lot more. We have now over the years surveyed what we call the fate space of early human development. In other words, the inner circles here. This is essentially to be thought of as a circularized lineage tree of the human organism, where you have the pluripotent stem cell sitting right in the middle. That is the ground state. No specialization. The perimeter that you have on this circle is all the cells that we consist of. This is the adult human. And the path to make them is essentially viewed here as you move from the inner center to the outer rings, through the outer rings, as you coordinate jump between the phenotypes of the progenitors and eventually land in the more mature cell. And what our technology allows us is that we can actually jump between these phenotypes in a rather systematic way. And the method is exactly the same regardless of what cell that we're making. And this is why we have a large opportunity to make cells across the entire human biology using this method. We make products now. We are seeking to build and invest in manufacturing, and of course we move in to production. Most of the data that we generate are obtained on plastic cultures. They need to be transferred into a scaling environment. There are different types of reactors you can use, some of which are shown here. The output is significant, and the PBS bioreactors as shown here are specifically useful because of the low shear operation. So our methods takes time to go from the research and development phase, where we can build the protocol up and then see it going through what is the design transfer process into ultimate manufacturing at higher volumes. And we want our quality elements to be solid, and we want them to exceed and extend beyond what you usually see. For instance, we would love and to simply set the standard that you never get an iPSC product purchased unless you also can download its associated RNA-seq profile. So you can take a look at what it is that you're getting instead of just hoping for the best, because there's too much batch-to-batch variation between these products in the current market. So, and to justify such a deeper quality control element requires a larger batch production and production towards inventory. So the products, of course, look like standard. They are cryovials, and we would produce to inventory. You would buy from this, and the cells are essentially received in the same way as if you received a cell from a competitor, with instructions for use, some media and supplements. And you can test these cells. We are very, very pleased to work with a lot of different clients and customers for offering the cells for testing, also for pre-release. So do not hesitate to contact us directly for anything you see on our webpage. There are many, many ways to contractually engage with us, both pre-release and post-release, and we have a very welcoming business development team to help you. As we go forward, we expect that we will offer cells in different formats, so it's not just the cells, it would also be packaged in kits, as some competitors are doing. That should be very helpful because it's an isogenic set, and you should be able to complete work with the set as to build either an organoid, another 3D structure, or say emulate biological systems such as the blood-brain barrier as mentioned here. We are already now seeing success taking disease-specific iPSC lines in, so that you can get pairs of normal and disease-relevant lines. For instance, the Parkinson's line that we sourced in actually from a European iPSC bank in this situation has been shown to be replicating elements of deficiencies within the dopaminergic neurons. We would also like to make fluorescent colored versions of the cells and offer them so that 3D printers can actually print with different colors or hues of the cells that they start with. We also have the ability, if you have already invested in generating an iPSC, maybe you've done some gene editing for CRISPR specific edit so that you are having the isogenic control and your edit, but have found a little bit problematic to actually differentiate them to what you want. This is where we can come in and we can receive your cells under MTA. We cannot, of course, sell further from that. All the material needs to go back to you. We would take these cells in, they'll undergo quality control on our side and quarantine, and then we will apply one of our protocols, and under the contractually agreed scaling output, we will then send the material back to you. That essentially then represents sort of a gap filling service, although it's more like a contract manufacturing service when you look at it. We're not a CRO. I have to say that we do not want to build the company as a contract research organization. We develop these protocols ourselves, but we are a product company, and this is essentially a way to sort of piggyback in on our attempts to build a broader scaled production for cells as products that we sell. So again, don't hesitate to reach out to discuss if we can help you there, so that we can get your biggest problem addressed with our protocol. We have built a facility now that is, we're very happy. We built it around the processes and the robotics and the tissue culture, and we're still expanding in this. We don't have currently any cGMP production facilities as example. We still need far more expansion capabilities from a production perspective, but we are able to do what we need to do in a rather cost-efficient manner here in Cleveland. So the question is, after the webinar, and I hope you will be interested in both what Reprocell offers and what Trailhead Biosystems offers, what can we actually do for you? I do think that you're here because you trust the brand of Reprocell, been around as one of the very first companies in the iPSC space, and has a breadth of offerings related to sourcing, the quality of generating reprogramming, and then also can provide a lot of other services within that space. We sort of sit here today to represent that gap, the gap between the reprogramming and then into a spatialization of cells. And as I mentioned, we focus internally on protocol development and cell manufacturing, but we do have different ways where we can engage with outside clients and partners. You have to view that the use of the cells is as flexible as you are in your thoughts. And so what are you using it for? Are you in a pharma company using the cells for drug discovery or drug safety testing? Or are you in an academic context wanting to get the cells for a specific disease understanding? Or it could be that a therapy situation demands that you have a very long-term perspective on clinical success. But in all situations, the front end of this should be relevant. The take-home message from my presentation today is essentially that our technology, HD-DoE, yes, it is mathematics, it's applied here to biology. It saves a lot of run cost. In other words, it accelerates the science needed. Well, we use the mathematical context of MVDA, which is baked into HD-DoE, and MVDA is multivariate data analysis. And it is from that we get these mathematical models of effector response matrices that I mentioned. And it is within that we can extract in a machine way, unbiased, these interactions that control the cell fates. And it's because we can identify the interactions that we have better protocols. And I think the biggest thing I can say is that when you gear up DoE, even biology will yield to that. So you now have comparatively built an experimental method that is approaching the complexity of the target that it is used at. And first and foremost here, both companies are very happy to engage with you in multiple ways. And that's my thank you. You can find out more about our cells and our programs if you send emails to info@trailbio.com and reach out, especially use LinkedIn. This is the platform that we're mainly active on. This is where most of the stuff is coming. You will find on our website a segment called Trail Notes. I may recommend that you take a look at that. There is actually a little written piece for me when I contemplate about the first decade with HD-DoE, but there's a lot of other things related to the biology, and that will be growing as we go forward. We will have our own webinar series as well, but we're early in the go-to-market phase, so we're very, very pleased that I get the opportunity today on the Reprocell platform to address you all. And the same thing with Reprocell. As you all know, there is a deep and comprehensive description of the services and activities that Reprocell can do for you.